a professor at the University of Maryland College Park in biometrics, and she retired in 2004. And she was my first biostat teacher and one of the best teachers I had. So in my long education career. Yeah, I graduated from College Park in 2003, right before she retired. No, she said she was an assistant professor when she taught me. And then after retiring from College Park, she went to the FDA at the Center for Device and was a team leader for diagnostic devices, and she did that from 2005 to 2010. Then she moved to the Center for Biologics, where she was the deputy director for that division from 2010 to 2011, and then moving on to being the acting and permanent director of the division from 2011 to 2016. And in that role, she worked on many guidances, including 2 for adaptive designs. And then she retired again in December of 2016. But she just can't seem to stay retired. So now she works at the Center for Drug Evaluation in the Office of Biostatistics, and she started that in 2017 and continues. And she's working as a senior advisor in an office group working on science and complex innovative science and policy. And in her own words, she said, "I'm working on safety, rare disease initiatives, and complex innovative designs, having fun working on interesting questions and no longer supervising anyone." And, and she is also a fellow of the American Statistical Association. So thank you so much for coming, Estelle.
Thanks for having me, and thanks for coming at this ungodly hour of the morning, which is fine. See, how do I advance the slides? Let's see, is this?
Yeah, yes, that should work. Yes?
Okay, so you get it. Okay, so today I want to talk a little bit about the importance of adaptive designs in a regulatory environment like FDA. And then I'm going to talk about something that I feel very proud of because it was my idea to begin with, and it's become a big initiative, is this Complex Innovative Design Pilot Program. So I thought, for those of you who don't know much about FDA, I should say a little bit about it. The role of regulations and guidances. Guidances are kind of neat because you can find them online, and they're kind of like a set of information that anyone can download, and you can sort of see what the FDA is thinking on stuff. And I'll talk about what an adaptive design is, a little bit about the Complex Innovative Design Pilot Program, a little bit about medical devices, because I happen to have worked on that guidance too, and the role simulations has played in all this, and then conclusions. I love this slide. Somebody gave it to me, and I just can't resist. Okay. There's a whole bunch of different centers at the FDA, and I happen to have been in all 3, as Janet sort of pointed out, and I will tell you, it's like different worlds in each of the centers. They have their own cultures and stuff. Every one of them has their own stack group geared to their particular statistics problems. And the ones that are medical are the Center for Biologics, which does vaccines, anything to do with the blood supply, and has become very hot lately because we've approved gene therapies and these CAR T therapies, which have taking patients who can't respond to cancer otherwise, to treatments otherwise, and basically give them years of extra life. It's pretty amazing, that stuff. Center for Devices and Radiological Health, the Center for Drug Evaluation Research. And to put things in context, they're such different sizes. There are over 200 statisticians in the Center for Drug Evaluation Research. There's about 75 in the Center for Devices. There's about 35 in the Center for Biologics. So we were, in fact, the smallest of the 3 medical centers. And Aura does a lot of the inspection stuff on medical products and making sure that what comes into the country is something that ought to come into the country and not. Okay. I recognize that a lot of you may not realize that academic trials do come to the agency. Sometimes they're actually required to come to the agency. In the Center for Biologics, because we regulated blood supply, there are products that would be used in the context of trauma, and the Defense Department would fund stuff. They still have to come and talk to us. They waive individual informed consent, but they're required to have community informed consent. And there are a whole bunch of guidelines about when you can do this and what you can't do with it, et cetera, and there's a separate guidance on that. When a funder of a study says, gee, I want Johns Hopkins to run a trial for me, but I want it on an FDA label, you really have to come to FDA to make sure FDA is okay with what you're proposing, et cetera. When the study involves an unapproved medical product, there are, if you notice, I have the word IND, that's investigational new drug. So for drugs and biologics, there are specific requirements out there in terms of when you have to come to FDA. And when you do a medical device, there's a different regulation that you have to follow in order to know when to come. And I write significant risk because medical devices includes things like in vitro diagnostics, and if all you're doing is taking a blood sample, they don't have to come to the agency. And my theory is that's probably why we get such messy stuff in diagnostics, is because they don't have to come to talk to us first. But if you're going to use radiation, even in a mammogram or anything like that, you have to have an IDE because they want to make sure you're not exposing patients to an excess. Safety is the word in INDs and IDEs and why you have to come overall. CFR stands for Code of Federal Regulations. And most trials at FDA are really coming from sponsors that want to market products that have medical indications. The two regulatory paradigms that we see are drug and biologics are in one space, devices are another. I was in CBER for 6 years. It's the only center that does a hybrid of both, devices, drugs, and biologics. And so we had a good position because we could say why the two elephants in the room need to talk to each other because we had to live with what both of them were doing. So it was kind of fun. Most diagnostics are in fact regulated as medical devices. Most therapeutics, but not all, are usually in the drug and biologic frame. And the development of the two are really quite different. And it's different companies, different sponsors. Sometimes the company will overlap, like J&J I think does both. There's a handful of companies that do both, but most companies are either mostly one or mostly the other. But the reason I think it's important is the statutes rather than the center drive the requirements. So when a diagnostic is used to decide who gets a new drug, you may have more than one center involved, and that's when you realize that the regulations are not the same and people aren't viewing things the same, but they've learned to get along and do the right thing. So the three centers are interested in total product life cycle. We see a lot more emphasis on post-market safety, and companies are constantly adding more indications, so there's no such thing as, one shot out the door. It's an evolutionary process for everybody. To me, the biggest challenge was the acronyms. If you've been here too, I remember at College Park it was really, so to try and give you an idea, oncology is by far the most challenging in some ways. We now have an agency oncology center of excellence. And all that's really done is taking the medical officers from the two centers that do drugs and biologics, and now they're under a separate umbrella. But I thought if I listed the various products and say, gee, they could be in one center or another center, it might give you an idea as to how complex it is, because we all have to talk to each other at some point. Basically, cancer vaccines, these new CAR T therapeutics, devices to isolate cells for therapies, that's the CBER product. Most drugs, and what do I call simple biologics? My colleagues at CITI go crazy when I say that. The monoclonals, single proteins, whereas in CBER it's the cells, immunoglobulins, things along those lines belong in CBER. Radiation devices, whether it's for diagnostic or therapeutic use, is in the Center for Devices. Cryotherapies, you can go on and on. When they have to do things jointly, they have to figure out how to split the responsibility. So if you have a diagnostic that says, this drug is for you, it has to go to the Center for Biologics, Center for Devices, or a diagnostic center. And then it also has to go to the drugs, and they have to learn to talk to each other, and it's not always easy. So submissions get a primary center for review. There's targeted therapies that involve a joint review. And each center, as I indicated before, has its own stack group. Epidemiology, this was my rude awakening, since this is largely an epi department. When I came to FDA, epidemiology seems to be preoccupied with post-market safety. And I think it's a missed opportunity myself. I wish epidemiologists were there at the design stage so that we did things that were sensible. We understood something about the disease, who was impacted, etc. But that's not the way it works at FDA. Okay? Guidances. Guidances are kind of important because they send a message to industry about what kind of things we're thinking about. So it's important that statutes, so the stuff Congress comes up with, regulations or binding interpretations of the law, they often go up to the White House and back to make sure we haven't done anything that's beyond what we're allowed to do. And then guidance documents and non-binding descriptions of FDA's current thinking on a topic. And sometimes there's a workshop beforehand. Sometimes you issue a draft guidance, and then you're shocked by the comments that come back, so you have a workshop. And then the comments are all received internally, and then we revise the guidance document, and then you see what's called the final guidance. And every few years you have to go back and revisit it to say whether it's really current enough. And my point is to pay attention to who's on the cover page. Why? Because we have two regulatory paradigms. It turns out we have two guidances for adaptive designs. One is for drugs and biologics, and the other one is for medical devices. I'm one of about 2 statisticians, I think, that have worked on both. When I was in CBER, CBER regulates devices, CBER regulates drugs and biologics, and I had to have my staff sit down and look at both documents and say, can you live with both in the same space? Okay? So basically, in the devices, I wound up writing some of the diagnostic aspects of it because most of the devices in CBER are in fact diagnostic, and when I left CDRH, the Center for Devices, I'd worked in every review division at some point. And so I helped with that section. The adaptive designs for clinical trials and biologics, I'll say a little bit more about that in a minute. But if you take a look down below, you see CBER is a co-sponsor of both of them. If you go to a different center, they won't hold that as being part of their message. And you hope that they're all on the same page and that they're all based on science, but you can sometimes see that a guidance could be issued by a division because it's focused on a certain class of products. Sometimes it's post-market safety. Sometimes all 3 centers are bought into the same guidance because they don't think there's an inconsistency, or for those targeted therapies, they know they all have to live in the same space. So they basically write a guidance together. So the CBER's, the Center for Drugs, Center for Biologics joint guidance, there was a new draft issued in September of 2018. It replaced the 2010 draft, in part because that draft generated 800 comments, including sometimes statisticians from the FDA. It got kind of awkward. The 2010 guidance kept talking about guidances as being, certain study designs as being well understood and less well understood. And language is really important because the guidance made it very clear. Well understood means we've seen lots of this at the agency. Less well understood said we had not enough experience at it. But both reviewers in my division and in other parts of the agency meant less well understood means we're not going to take it. There were people in industry who stopped the statisticians from submitting stuff because they said FDA won't take it. So we decided instead of binning designs, we would write a guidance that was based on principles, which is a lot like the CDRH guidance. It expanded discussion on technical aspects such as estimation, simulation, and Bayesian methods. And it added some clarity on what FDA is going to ask in the context of review. Okay. So first of all, what is an adaptive design? You guys are in a program where I'm sure you have some idea, but FDA's got a definition, which I think is pretty consistent with the literature. A clinical trial design that allows for prospectively planned modifications of one or more aspects of the design based on accumulating data from subjects in the study. It's not unplanned changes. We're not looking for ad hoc stuff in the middle of a study that calls into question trial integrity. And there's lots of room to cheat, and we really want to keep an honest trial with an honest conclusion. The protocol amendments are based on information from sources external to the study. Sometimes you get surprised. Sometimes the same product is approved abroad, and that changes the whole perception of who's willing to come into the trial. So there always have to be discussions. It's not because the company's cheating. There are all kinds of things that could cause an issue, and they have to be evaluated on a case-by-case basis. Okay? So the motivation for adaptation is, in theory, it should make you statistically more efficient. It's very rare that when a student or a company is planning a study that they know all the things they need to know to plan the study. They may not have a good handle on variance. They may not have a good handle on the control group. They may not have a good handle on a lot of other things. So the idea with statistical efficiency is maybe you'll learn something partway through that you can incorporate into improving the design, including changing the study size or potentially stopping early, either for you're never going to make it because it's stopped for futility, which is actually a very positive thing given the high cost of trials, or stopping for success. We may see better improvement of understanding dose-response relationship with adaptive dose selection. There's lots of options. The limitations is the opportunity to cheat. You know, you want to be honest. Sometimes people don't even realize they're cheating in the sense that they don't realize what the biases are. So it's not about me saying, oh, somebody's being corrupt or anything like that. I don't want that to be there. But there's operational challenges. If you're going to change how many individuals are in each arm, do all the clinical sites have the right supplies of all the right drugs? I mean, sometimes there's a logistics issue that you want to do. The other thing is when you start changing the amount of each, as long as they're appropriately blinded, you're fine. But if you wind up unblinding for whatever reason, then you can sort of say, well, what message is it sending? So FDA draws a lot of distinctions between exploratory studies versus the stuff that's going to show up as support for going into a label. So they encourage companies to actually play in early space as long as it doesn't violate safety considerations. Studying doses and different combinations and formulations and stuff in an early phase design is something you really want to do. You don't really want to do that in a big phase 3 study. And in vaccines, where accruing patients is very easy in vaccines because it's healthy people, in theory, the late stage study is quite big. And you really wouldn't want to be screwing around with different kinds of doses and different formulations and timings and stuff like that in a late stage design. You really ought to have your act together and do that in an earlier stage. And then you're running the pivotal study on everybody. Accumulating study data, we draw a distinction between blinded and unblinded kinds of analyses. By blinded is you can take a look at the data and you go, I don't have enough events across both arms. You know, you don't really have them broken down by arm. You sort of careful about how many. You say, I'm never going to be successful because some of the survival analysis methods, for example, are event-driven, and I don't have enough events to tell me much of anything. Maybe I need to go get more subjects just to have the event rate, because medicine is changing and sometimes the patients are doing better now than you would have anticipated using a study that was done 10 years earlier. So there's always some of that. There are types of adaptive designs. Sometimes the adaptation is based on who you recruit to the study, so based on characteristics. Sometimes it's based on pooled outcome data. And then there are ones that are based on comparative data. And I'll say a little bit more about group sequential designs that go along, but you can adapt the sample size, you can adapt the population, that's the adaptive enrichment stuff. You can adapt by adding or dropping arms, depending upon what you find. How many patients go into each arm is patient allocation. And adaptation to endpoint selection, I would say that that's very tricky. You really have to have that resolved kind of early on, or I can imagine that there would be problems. Pooled outcome data, sample size depends on significance, power, nuisance parameters. Nuisance parameters, for those of you who are not statisticians, are what we call variances or event rates on a control arm. They drive sample size, but they're not exactly what you're interested in doing when you're done. So the goal is you can use accumulating information about variances to modify sample size. I had a grad student who's actually a full-time statistician at FDA that tried to look at adaptive designs for bioequivalence studies. They're much smaller. You can't rely on asymptotic theory. And this was co-advised with Eric Slud at College Park. And so I'm hoping that that's kind of exciting. There's lots of room for doing things. And because it used pooled data, there was no concern over bias. Case studies. These are case studies that are in the guidance. So don't ask me about the details about the particular drug, though some of these I've actually worked on. By looking at stuff for a treatment for patients with severe head injury. There's a 3-category outcome defining functional status as 6 months. They weren't sure where the control arm sat with respect to these kinds of outcomes. So the interim analysis allowed people to update estimated proportions based on pooled non-comparative data, and they increased sample size with no alpha penalty because it's pooled. And they avoided inadequately powered design. They upped the sample size from 400 to 450, and they were successful. Okay. Group sequential designs have been around for decades. You can stop early due to it's a good thing because it's efficacy, it's a bad thing because it's futility, or there's harm or safety that you hadn't anticipated. The trial participants don't have to hang around longer than you need to reach a conclusion. Individuals outside the trial are provided more effective treatment options sooner. There's maybe economic benefits, and they're relatively common at the FDA. But one of the things I will point out is there are 2 things that are in its favor. In group sequential, you don't have to tell anybody you're stopping till you stop. Whereas if you decided to plan small and add subjects later, there's a message that's being sent. So when people were playing around with some of the newer adaptive designs, that is a concern that does still remain. The other thing that I think is important is when you stop early, the effect size could be exaggerated. It's like a random high kind of thing. So the case study that they cite in the guidance is one for heart failure. There were interim analyses. Now, you see, with group sequential, what we tell the company is you can pick the interim analyses you want, okay, but we want them in the protocol so that we know that you've done it on time, etc. Even the Land-de-Metz approach for adaptive designs says you'd be flexible. We still want companies to say when they're going to look, only for trial integrity reasons, nothing else. This one stopped with 2/3 of the data, and so company saved a lot of money, okay, and the product was approved. There are other types of adaptations based on comparative interim results. We have taken some based on the promising zone approach. The idea of a promising zone, I think Cyrus Mehta wrote the original papers dealing with this, is you get to a middle stage and you have this planned look, and sometimes you say, oh, there's no way in hell this is going to work, so, and this is all on tape, right, Janet? So you stop for early, for futility, or you stop for success and you say, I'm not going to be able to tell where I am, I need more data. So the promising zone is like the sweet spot where I'm going to go ahead and augment data. Adaptive enrichment. We've actually funded 2 projects with Michael Rosenblum from your biostat department in the adaptive enrichment phase. They're not easy analytically. I think when you see the results from each patient rather rapidly, it's easier to enrich. But the real interest in adaptive designs is in these long-term studies like oncology or other stuff. Sometimes you won't see the outcome for a while. How are you going to adapt? So the theory gets much messier because you need to say, well, are there biomarkers that might give me an indication of how I might go about adapting, even though it's not the primary endpoint of interest? Treatment arm selection, you might want to figure out what the right dose is. And sometimes companies combine a dose selection phase with the actual pivotal study. And in some therapeutic areas, it's called seamless phase 2-3. The new guidance tries to avoid that terminology because different therapeutic areas have different ideas of what a phase 2 trial is like. So they just finally decided they would talk about what it is you're trying to achieve with a part of your study rather than calling it that way. Play the winner. I happen to be friends with some people at NIH, and Mike Proshan, who's a very smart guy, and these play the winner studies, Mike Proshan goes, remember the ECMO. There's a very famous study that I saw when I was a grad student, so it was a long time ago. It's an oxygenation type therapy for premature infants. And Marv Zellin, who's a very famous biostatistician, who's not around anymore, came up with a study where you looked at the first patient. If the patient does well, you stay on that treatment. That's called play the winner for the next person. Now, what happened in the initial ECMO trial is the first patient died, and they were on the control arm. So they then switched them to the treatment arm. And the next 8 patients did well on the treatment arm. But at the end of the day, you only had one patient on the control arm. So play the winner has a problem. You need enough data before you can intelligently decide that that's the way you want to go thing. It's not a good idea to do that adaptation too early in the game because you can wind up in that kind of a mess. So you have to be careful. And adaptations to endpoint selection, I haven't really seen much of that, but it is mentioned in the guidance. I think you would want to do that early on. What companies often do, if they think there are 2 endpoints to win, they do an alpha penalty. They decide how much alpha they're going to allocate to each endpoint and go forward, and that's typical. And one of the things they do at the agency, which you may not see in an academic publication, is they do hold people to type 1 error control. It gets more entertaining with some of the Bayesian submissions, but that's where we are. This is one I worked on, okay? So I can say a little bit about this. This is an HPV vaccine. It's 2nd generation, and that became really important. It's a 9-valent HPV vaccine. If you get the vaccine, it helps you avoid certain forms of cancer. It's been approved for women for cervical cancer. It's been approved for both genders for other types of cancers that HPV is associated with. What was allowed here was the ability to take data from the phase 2 part of the study and include it in the pivotal study. The reality is, I will tell you, these studies are often overpowered in vaccines because you can't anticipate where the cases are going to come up. And what drives efficacy in a vaccine trial is showing the number of cases goes down as a result of the vaccine. So it was a huge trial, but they used biomarkers to select which dose they wanted. They had immune markers, which are very common in phase 2 trials and vaccines, to decide which dose they wanted to carry forward into the late phase study. And this was a multi-regional study. If you notice, I have a line at the very bottom. One of the things that I did when I was division director is one of my statisticians went through all the INDs we had, all these investigational ones, and identified all the ones we have with adaptive features. And Therapeutic Innovation and Regulatory Science, it used to be called the DIA Journal, has got a survey of the CBER results that we had. So we showed people the kind of adaptations we were typically seeing, so people got a message of what was common and what was not. But the reason I mention that is there's a nice discussion of the HPV vaccine in particular, and the Office of Vaccines likes having a conversation with companies at the end of phase 2. Before they're going to expose 13,000 patients to a vaccine, they want to make sure they're comfortable on the safety, they're more comfortable on generally what's happening, and they don't want to intercede too much. So on the 2nd generation vaccine, they were more comfortable with foregoing the end of phase 2 discussions and allowing the company to do a seamless phase 2/3. So I think the point is it may not be statistical issues solely that drive the decision to allow certain types of designs. It's a question of whether they want to see the results and come and talk to you about some considerations. If there were safety issues at the end of phase 2, they might say you can't take that dose to phase 3. So I guess I wanted to point out it's not purely formulas. Stampede, this is in the guidance. I wasn't 100% sure we saw this at the agency. This falls into this class of multi-arm, multi-stage adaptive designs. I believe it was done in Europe, and it looks at multiple approved therapies for prostate cancer associated with androgen deprivation therapies. It's what they call a platform trial, where you're looking at lots of therapies all at the same time, trying which ones will work and not work. We see a handful of these at the agency, and I'll say a little bit more. But in comparative effectiveness, this could be a very logical thing to want to do, is to put a lot of treatments on the same study and decide how you're going to add and drop and decide what stuff is worth retaining. You have the advantage of a big umbrella trial. They get to share control arms. So there could be some logical stuff. And we work with companies in this platform design space. We have particular master files we can put, and then we have individual submissions. So companies have to decide what things go into the master file versus the individual application. Okay. Complex adaptive designs. They can include multiple types of adaptations. Things get less predictable the more stuff you try to twist in your trial. It could be also adaptations to the answer man question, that would be the enrichment studies, for example. When you go down to a population, you change your definition of treatment effect. What are you going to do? And you invariably wind up with simulations. I happen to be of the belief that even the simpler designs could stand to have simulations, because I think clinicians, when they sit down with you, if they see individual trial results, they develop more of a feeling for what the trial is really doing. And they might say, that's implausible, that's plausible, etc. So I happen to like simulations a lot. But in this case, you can't, there are a lot of things you can't do without simulations in some of these. The PREVAIL study, I happen to work on this too. During the Ebola crisis, it was a madhouse at FDA, at least with respect to me. I worked on, I was in the office that dealt with Ebola vaccines. And finally we had the unfortunate situation of being able to test the vaccine in the real world. It was done in Africa. But there were also therapies. And a platform trial is one that allowed for different treatments that could be tested. And we realized the sample size would have to be fairly small. People are not excited about being randomized to a control in the middle of an Ebola crisis. So one of the big debates was the role of randomized trials. And we felt the only way we could get an honest answer on some of these things was with a randomized trial. But the idea is that patients in the study would still get the best available standard of care. And in Ebola, that actually makes a difference. Fluid management, the types of things that you do with patients can really make a difference. But if you notice, the interim analyses were really small. Mike Proshan, who designed some of this stuff, called it his barely Bayesian design. There's a new opportunity to bring new things on as time went on. There's a special issue on Ebola in the Journal of Clinical Trials in 2016, where you can see various study designs that were proposed. I was part of an opinion piece that came from FDA. And the EMA weighed in. That's our European counterparts, et cetera. So it's an interesting place to go look for some neat stuff. So the thing that regulators worry about is they don't want type 1 error inflated. That's a fancy way of saying we don't want the wrong conclusion. Our regulations say you're supposed to show the stuff is safe and effective, and if nothing is going on and you still conclude it's safe and effective, that's a problem for us as regulators. You can't be perfect. You always know that there's measurement error and stuff. That could be a problem. The only alternative to never being wrong is to have study sizes that are in the millions, and I don't think we're going there. We want treatment effects that are reasonable if we have to judge risk-benefit. We want the details of the design to be appropriately pre-specified for integrity reasons, and obviously we worry about who knows what and when they know it in terms of maintaining trial integrity, so we definitely like blinded studies anytime we can. We're very concerned with the adaptation plan, the monitoring, who sees what, when do they see it, et cetera. And when they come in, we ask for the steering committee minutes or the data monitoring committee minutes, if there's a data monitoring committee, and make sure that stuff is appropriately reported. Okay. When to adapt. Trial decision depends on many factors. I tell companies, play. Do your simulations, do your analytical work. Think about simpler designs in relationship to complex ones, because complex ones means you've got to worry about the supply side, you've got to worry about who knows what and when they know it. Make sure you're getting your money's worth out of doing an adaptive design, because there's a lot more upfront planning for it to make sure things go right. The worst thing that you do with a very fancy adaptive design is something goes wrong, and now what do I do? You may have to start the whole thing all over again, and that would be horrible. So what happened in 2016, two things happened, okay? This complex innovative designs, there was a piece of legislation that got passed in the late 2016 called 21st Century Cures. And it called for more use of complex innovative designs. Of course, I had to define it because we had discussions, and I said, if you have to use a lot of simulations in order to figure out its operating characteristics, I'm going to call that a complex innovative design. Because there are many people who felt like when you trained in grad school, you taught these nice theoretical proofs, closed-form formulas for everything. We're saying not everything can be done with closed-form formulas. You may have to do simulations to convince us type 1 error is reasonable, etc. So that was the generic definition. Bayesian stuff is not in the 21st century cures at all. I think, well, it's on tape. Some member of Congress basically said they thought that was lowering the standard. I don't believe that. I believe there are Bayesian analyses that are good. There are Bayesian analyses that don't make sense. There are frequentist analyses that are good. There are frequentist analyses that don't make sense. And so you need to think through what you're using and why you're using it. But we negotiate with industry every 5 years on a use of fee drug act. Companies spend a lot of money to come into the agency. There are some exceptions for orphan diseases and small businesses, but there are big ticket fees that are given to the agency, and it pays my salary. So we still get substantive amount of money from Congress. Don't, you know, this is not the sole budget, but it makes the difference in terms of being able to hire people and get the staff we need and turn around stuff. It comes with a price. You have to have, there are clocks that we have to meet. You know, when are we going to get our decisions and how are they negotiated? But what came up with industry is another thing that I talked about was this complex innovative design pilot program. And Lisa Lavange, who at that time was in charge of drugs and I was in biologics, I said what really got me upset was going to a meeting and say, yeah, we've allowed adaptive designs and I'm not going to tell you what we did. We'll keep it a secret. We're required by law to keep negotiations with companies confidential. I couldn't talk about the adaptive design without naming the company, and that got kind of awkward. So we have a voluntary program, that's the CID pilot program. Companies can come in with a proposal. They have to have a drug on the table because this isn't about academics coming to us with novel designs. They have plenty of options about doing it. This is about a company coming in under an IND and saying, this is the kind of design I want and the context for what I want it. And there's negotiations with lawyers. What can we say in public versus what we can't? But the idea is once we pick something, we have the ability to learn collaboratively. It may very well be that we might take a certain design if the following situations are true, and we could sort of say, here's why it makes sense here, here's why it doesn't make sense here, to give industry an opportunity to think through the same things that we're thinking about. So public forums could mean publications, it could be workshops. We're still in the relatively new part of this. But complex innovative designs, many of the adaptive designs can fall into this bucket. There are other innovations that are possible that I wouldn't strictly call adaptive designs, but as I indicated, simulations are often required to understand what's going on. So one of the areas where industry is very, very interested is what kind of other data can I leverage in my decision making? In rare diseases, placebo arm may be unethical, but we may have well-conducted natural history studies. Registries are kind of interesting. Some of them include natural history data, but some of them are hybrids from previous trials. Some of them are, I'm not sure what they are. Epidemiologists could play a huge role, in my opinion, in getting best epi practices into a registry in terms of what gets collected, why, et cetera, and how it reflects standard of care for those subjects in the absence of an active treatment. But the big statistical question is, when should we borrow and how is still a question. Could we leverage some borrowing and some not borrowing concurrent controls in order to maintain blinds, et cetera? We're still in the throes of looking at that stuff, but that is one of the things that would fall into the CID pilot program. In the anti-infective drug development, we've already had workshops downtown. Right now, when an antibiotic comes to FDA, if that pathogen causes a lung infection, that may be a different study than when the pathogen causes an abdominal infection. And the question is there a potential for borrowing, and do we understand when and where? And that would lead to some kind of a Bayesian approach. Tom Lewis is now doing an IPA at FDA. There is life after retirement, I guess. And this is one of the areas that he's been asked for his advice on. He's working with the anti-infectives group in particular, so I'm sure it'll get well done, whatever they come up with. Platform trials. The original platform trial at FDA is probably something in vaccines in the 1990s, but the initial platform trial that got a lot of hype was a phase 2 adaptive design trial in oncology called I-SPY 2. And I happen to have been the medical device reviewer at the time, dealing with the diagnostics in the trial, not the therapeutics. So, but platform trials are one trial, multiple goals, and possible sharing of control arms. So it sounds appealing, but companies don't always like it. You know, it's like, it's not FDA. Companies may not want to be in the open competition. They may not be ready at the same time that the other company is. So there could be lots of reasons why it doesn't work, but there have been a few. In cancer, there's a lung map study. It's been around for a while, but the Friends of Cancer Research announced this huge expansion because how you treat lung cancer involves a lot of biomarkers, because there are lots of different cell types you have in the lungs, and they discovered different treatments may work for one kind of cancer subtype or not another, and lung map involves both biomarkers and treatment. The STAMPEDE trial that I mentioned for prostate cancer is, I'm not convinced there were new drugs on the table, but if there were an interest in getting into some labeling kind of stuff, FDA could get involved. The PREVAIL study, as I mentioned, but there are others coming along. There's a really nice article that Alyssa Lavange and Janet Woodcock, for those who don't recognize names, Alyssa Lavange used to be the chief statistician of the Center for Drugs. Janet Woodcock is the head of the Center for Drugs. And they wrote an article in the New England Journal of Medicine, I think it was January of 2017, that talked about platform trials at the agency. Umbrella trials, it's different drugs for similar indications. So the I-SPY 2 happened to be for neoadjuvant cancer. Obviously, lung map involves multiple drugs and multiple biomarkers. Basket trials could come from the same company. Why? Because right now you might need a different trial for each type of cancer. And you could decide, I want this drug and I want to test it upon a bunch of different cancers, but then you can't change control arms because the arms have to have the same cancer. But there may be some advantages with one overall management of a trial. Some of the non-stat issues may play a role. Autoimmune diseases are another one where the same drug is used across multiple indications. So basket trials have the advantage of one company can come in and say, I want to run a basket trial, and it's their decision. There's potential use in comparative effectiveness outside of FDA. NIH and FDA went together on a project called ADAPT IT, where Berry Consultants won the contract, so you know it's going to involve Bayesian submissions for anybody that sort of plays in the space. But I remember one of the studies was really a comparative effectiveness trial. They were looking at treatments for neurological injuries in the emergency room. And I'm sure it's been published, if I have little doubt. So comparative effectiveness, where you have lots of treatments that are potential and you want to figure out which might be best for this group of patients or that kind of patients, you could potentially decide you want to run a randomized trial in that space and use a platform trial. Medical devices. The adaptive designs for medical device trials, they're very similar, I mean, in a lot of ways. The product life cycle is very different from drugs. Devices evolve. It's one of the reasons why there was a Bayesian guidance in medical devices, though their evolution on where it fits in the regulatory paradigm has also changed. But companies would change the software algorithm deciding when to shock a patient for a defibrillator, because that's a medical device. You want a trial. The question is, how much more data do you want? And Bayesian borrowing might provide some opportunities, certainly in the control arm or something along those lines. So there is a guidance. It's unique to devices, and CBER signed on to it, by the way, but I wasn't in CBER yet, so I can't take credit for it, okay? But a lot of the device guidances involve what they call non-informative priors in the Bayesian space. But I've been told that they're interested now in doing more with informative priors. But some of that is because they have a lot of devices that are very similar, even from the same manufacturer, different models, et cetera, so they're able to make more direct comparisons. Devices evolve. They may have short life on the market before something else comes along, and a lot of studies can't be blinded. The surgeon knows what they're putting in. You know, it's just, you can't do perfect blinds, and so you have to decide. The CID pilot program is only for drugs and biologics. That's because of the way it got negotiated with industry. Devices has its own programs to promote innovation. I don't want to ever imply that they're not being innovative, because in some ways they push the Bayesian envelope. They've also done more with single-arm trials because they can't blind. So I'll say a little bit more soon. But, and they're also part of this medical device innovation consortium. So they have lots of other ways of promoting innovation in the device space. It's just not the same vocabulary as the CID pilot program. The adaptive design guidance builds on the two previous guidances. I didn't work on the Bayesian guidance, but I spent a lot of time on the pivotal investigations guidance, even though I was in CBER. I worked on the diagnostics, but I actually worked on the general clinical trial stuff that's there. They dealt with things like, you know, if you're going to do a surgical device, you've got to pay attention to how you're going to train the surgeon and things like that. So a lot of that stuff is in there. But what I particularly like about the pivotal investigations guidance, it talks about principles that might allow you to avoid a control arm versus when we really want to see a randomized trial versus when we might do an objective performance criteria. In objective performance criteria, there are a lot of devices that I would call nth-of-a-kind devices. And you basically run your study, you show that 90% do whatever you call a success, and you're out the door, or that's the lower confidence bound that you have to show. There's use of historical controls because the industry and medical societies foster use of registries. C.G. Wang, who's works with both Gary and Brosner and people in Biostat, he has a grant looking at some of this kind of idea of developing an objective performance criteria using registry data. So people are looking at different ways of analyzing the data. Historical controls, CDRH has done an awful lot with propensity scores to try and adjust for imbalances in old and new data. So there's opportunity for innovation at the agency, even though it's not always randomized designs. Diagnostic devices, which I worked on. The reality is most of the diagnostic device studies are not randomized, and truth involves a reference standard. You have to say what is truth, and I will tell you sometimes that's a big, curious debate with sponsors about what's considered a good reference method. The pivotal guidance talks about diagnostic performance studies. I don't remember us giving it a name until then, but that's what we did. The biggest concern I usually have in diagnostic devices is, do we have the right patients in the study? Spectrum bias. They love going to places like Hopkins, Mass General, etc., to get their patients, and you go, those are more seriously ill. They're referred to from other places. So we may very well tell a company, you got to add some community-based sites because that's who you want to sell to when you go post-market. We want to make sure there's no surprises. So spectrum bias, we have to pay attention to where they are. That tends to be a much bigger concern than a lot of the other things I've seen. We allow adaptive designs, but it would be kind of unusual in an in vitro diagnostic. But sometimes the diagnostic is something that has to run during surgery. And so the principles associated with adaptive designs still apply. So you got to think about the context about whether it pays. I have an ethical perspective on adaptive designs. I don't want patients exposed to something that's not working. And so I can sort of see an adaptive design for anything that puts patients at risk during the study as being something important. Okay. Simulations. So the innovative design pilot and adaptive and Bayesian elements in a clinical trial placed a greater emphasis on simulations, and it's not unique to drugs or devices. Operating characteristics could include type 1 error and power considerations. Bayesian gets trickier because what do you mean by a type 1 error if you have an informative prior? It's not immediately obvious because in a sense informative means you sort of sway the results a certain way, but you say, I can believe that. So it depends upon the kind of study design, but we have to think that through. I'm not sure we have all the answers in that form yet. Model choices should not be driven totally on the simplicity of the calculations, but what is needed. When I was a graduate student, which was a very long time ago, conjugate priors in the Bayesian world was the way I got exposed to Bayesian methods. Okay, and that meant the formulas were nice, closed, easy. The appropriate terms in numerator and denominator would cancel, and you could get a nice closed form solution. They're probably unrealistic most of the time. Not all, but most of the time. Subjective priors would not be accepted in regulatory study. More than likely, they want priors from empirical data that's collected, that's comparable to what you've got in the current study. So Bayesian computations are hard. You better know your MCMC methods. You better know what to run, when to run it, and so it gets pretty challenging. And reviewing these is really hard. One of the reasons I also pushed for the CID pilot program was because I had reviewers pulling an all-nighter when a company came in with something really complex, and I couldn't sustain that if the numbers of studies that came in that had that. So the only thing I wanted to say before I got done is that there are opportunities for research in this environment. One of the things that 21st Century Cures talks about is this notion of real-world evidence. That could include the natural history studies for disease, but one of the things in the 21st Century Cures is they say after you get your first product approved, if you want additional indications, there is a potential for using real-world evidence. We're still trying to figure out what that all means, but what kind of data could you use? And so there have been workshops downtown to look at this kind of stuff. Will insurance records be good enough, or electronic health records? What other things would we need to know? And obviously, as epidemiologists used to observational studies, there's a lot of place for doing something interesting. Enrichment using signatures. What do I mean by a signature? It combines multiple traits to decide how to enrich, not a single assay or something like that. Best practices for rare disease studies, including very tiny studies. Adaptive pragmatic trials. Technically, if a trial is randomized, it could be a pragmatic trial. I have not seen too many cluster randomized trials. I kind of wonder if we could be doing better in that space. And so there's a lot of room for doing things like that. Comparative effectiveness trials that are adaptive. I haven't seen very many of those. One of the challenges that came up in the NIH study was, how do we get these reviewed at NIH in the study sections? Do we have the right skill set at the table to really review an adaptive design given how much energy it takes at FDA? I'm not sure we're quite there yet. Methods to ensure trial integrity, best practices for reporting treatment effects in an adaptive design. As I pointed out, in group sequential, the effect size is exaggerated. But of course, there's meta-analyses where some of the designs are adaptive. I think you need to think about bias. You need to think about handling of missing data and a whole bunch of other things. There's a lot of potential in that space. And so my conclusions is not all the variations on adaptive designs have been used at FDA. We're amenable to new options. Some designs may be better fit for goals outside the agency. FDA has to approve products, and once they're safe and effective in a given context, and some options may be better for some settings and not others. And that's it. So hopefully I kept it to a reasonable time frame. I don't know if there's any questions I could answer.
I was wondering when you talked about the adaptive design being secret, that you couldn't share what the change, you know.
Well, you do change. The people who have to know it, it's a question of how much they know. In other words, the reality is they're going to know. So suppose you have to suddenly up the sample size by 40%. Then everybody's going to know you didn't get the effect size you proposed up front. And so that sends a message, you know, maybe investigators aren't so crazy about randomizing patients to your study anymore because the effect size isn't the kind of effect that you implied it might be. So that's really the thing that I worry about is how that gets affected in terms of the message. It's just like I said, things can change because something's approved abroad. And you're running the study in the US, and they go, gee, it's already effective, why should I randomize? You know, or maybe it's ineffective abroad because you knew something over there that you don't know here. It does affect people's behavior. And so you do need to think about the consequences, about who knows what and when they know it. So I think that is important. Data monitoring committees may also play a role in adaptive designs. We don't require them at the agency. NIH does, but we do not. We require them when informed consent is waived, but we do not mandate they have it. Doesn't mean it isn't a good idea. It convinces reviewers that the integrity is higher because you had somebody external to the company watching over you in terms of trying to decide when to adapt, how to change things without being influenced by what the company wants you to see. So, yeah, there are different things that come into the adaptive design space. DMCs are a good idea, but we wouldn't want the same people deciding which adaptations ought to be there and making the adaptations, because you want to stay objective. That's really the key. Any other questions I can? The data inside trials is usually better than the data outside trials for safety. It depends on the kind of events that you're looking for. Sometimes what they've done is they still think the risk-benefit is there because there's nothing else out there for these patients, and the patients and their physicians can decide if the risk-benefit is appropriate. And they may say there are additional burdens you have to come in with post-market to address some additional safety concerns that may come up. So in diabetes is an example where they now actually require additional data to demonstrate that there is not an elevated risk of cardiovascular problems as a result of taking a medication to improve your hemoglobin A1C for diabetes. Because there was a meta-analysis done in 2007 in New England Journal of Medicine that triggered all kinds of issues. And so far from what I can tell in all the studies where they've required it, they're not seeing the elevated risk. And the analyses were criticized a bit because of the way they handled studies that had no events, et cetera. So it generated a lot of stat papers and nothing else. So there are certain areas where they will require you to address safety in a pre-planned, control the alpha type fashion. Most of the time, safety is somewhat exploratory because it's hard to capture in a single variable. Think about challenge from a statistician's perspective. Do you want some liver issues, or do you want all of them grouped as a group? What are you going to analyze? So it winds up being a little bit more exploratory in flavor. Obviously, if patients die during the study, there's going to be a lot of attention paid. But some of the illnesses are very serious. So you're going to have to look at risk-benefit. You have to, something that's for dermatology and it's cosmetic, you don't expect deaths in the trial, and you won't tolerate it. Go ahead.
My question is, how often the adaptive design being used compared to the phase 3 trials versus phase 2 trials? From my understanding, it's more likely to be used on the phase 2 trials for dose escalation and dose selection versus the drugs being approved in the clinical trials using adaptive design.
Well, the very nature of some of the earlier phase studies is they're designed to be exploratory. If you don't do adaptive something to sort of choose what you want, I think you've missed an opportunity. In phase 1 in oncology, there's these CRM models, continual response. What?
Reassessment.
Yeah, so continual reassessment models, and we see them a lot in oncology. In biologics, maybe a little bit less for the cell therapies because dose is sort of harder to get very exact. But there's no question in the drug environment that CRM models are used a lot. I don't know how often I've seen it in some of the other therapeutic areas. One of the things that I've noticed in oncology tends to be a lot more innovative in some ways. It doesn't mean the other areas are not, but just oncology is hard to study. And I find that a lot of the adaptive designs we've seen have been in the oncology space. Phase 2 should be about dose exploration, so that's exploratory. And there's a tool that FDA actually recognized called MCP-mod that was developed by statisticians at Novartis in Europe. And it just suggests an approach to maybe selecting doses that may be a little bit less loosey-goosey, you know, just with some rigor. And the agency likes it, you know, but it doesn't mean that that's the only way you would go about doing it. You have to think about not only dose, but how often you give the dose. Does that matter? And the way you formulate it may make a difference. And a lot of that stuff tends to be done in the earlier phases. And some things can be done rigorously, but some things, in order to do it rigorously, you need a very big sample size. And so you find that companies are not that willing to run mega trials in early phases. It varies with the therapeutic area, and I don't always understand the business model, but it is what it is. Any other questions? Gary?
So, in the Cancer Center, we review protocols for science as part of our mandate to be a comprehensive cancer center. And we see a lot of drug company studies where companies participate. And it's not unusual to see a protocol where in the statistical consideration it says that a formal statistical analysis plan will be provided at some point in the future prior to data lock. And sometimes they don't even give information about boundaries that there will be interim analysis. So does that mean that somehow that's got through the FDA?
Oh, no, we get the same issue with the FDA. And I was working on this ICH guidance group dealing with estimands, and one of the emphasis in that one is tell me how you're going to deal with all your messy stuff, you know, and plan for it up front. But that means you need to be able to go back to the protocol and say, is the right information being collected? Do I know why a patient's leaving the study? Do I even know that it's really tolerability of the drug? Do I know other things? And basically what we heard from our medical officers is they're not ready for the final stat analysis plan when they're doing the protocol. So there is a gap. We do get concerned. We want the stat analysis plan before there's any unblinding at all. So that's, it becomes sort of like a regulatory compromise. I personally would prefer the stat analysis plan, or at least the draft of it at the time the protocol is being looked at. When I was a reviewer in CDRH, because as a team leader I also did reviews, I would line up the case report form, the protocol, and the stat analysis plan and say, are they in sync with each other? And it sounds like a really dumb thing to do, but sometimes somebody's proposing analysis of variance, I go, this thing doesn't have numbers. What are they doing? You know? And so I think that you should be asking for a draft, whether you see a final one or not. I think it's inherently stupid. You need to know how many patients you're putting at risk.
There's no guidance that we can point to.
I'll take a look, but I don't think there is. The E6 guidance spells out what needs to go into a protocol. That's an ICH guidance that's good for multiple regions, and it got revamped. You can take a look to see if it tells you what to put in a protocol. It might make you a little happier. It still might not give you, you know, the final stat analysis plan. I can ask around when I get back to the agency and forward things that I think could be guidances that may help a bit. But
Thank you very much. If there are additional questions, please come up. I think our appointed time is
over.
done. So thank you again,
No,
Estelle.
no
It was
problem.
very interesting.
Okay.
And appreciate you making the trip.
Thanks.